Okay, so assalamu alaikum everyone. My name is Ibrahim. I'm a second year medical student, and today we're going to be learning about erythrocytes. I've seen the group. I've seen you guys complain about this lecture. So inshallah, we can simplify it today. I've tried to include everything so you guys can refer to the slides or just use the PAL session. And I think that's it. Inshallah, if you guys have any questions, just send a question in the chat or open your mic. Okay, so. For the objectives of today's PAL, we have erythrocytes, erythrocyte membrane, the metabolism, and the metabolic effects. So, can anyone tell me what makes up the blood? The blood is made up of three layers. We have the plasma, we have the buffy coat, and we have the erythrocytes. The plasma is the liquid part of the blood, and it contains proteins such as albumin, electrolytes, and dissolved gases. The buffy coat has platelets and leukocytes, which are white blood cells. And finally, erythrocytes, which is our main topic for today's lecture. From this slide, I don't think you guys need to know much, but we have been asked before about albumin being in the plasma. Now, just an overview of everything before we get into the main bulk of the lecture. RBCs start out in the red bone marrow through the action of the hormone called erythropoietin. It is produced from the kidney and it acts on the red bone marrow. This process is known as erythropoiesis. The RBCs don't have a nucleus, they don't have organelles, and this also includes mitochondria. Because of this, they rely on anaerobic glycolysis for energy. Once they carry out their function and transport oxygen, they go to the spleen or the liver and they get taken up by macrophages. So this is just an overview, okay? Hold on. Okay. So this is everything I mentioned put into words, that erythrocytes are produced in the red bone marrow through the action of erythropoietin. And the doctor mentioned something in your slides, so I decided to add it. Let's say someone has hypoxia. Hypoxia is inadequate oxygen supply to tissue. The kidney will detect this low oxygen supply and produce erythropoietin as a response. But because it's producing a lot of erythropoietin, it acts on the red bone marrow, and as to compensate for the low oxygen levels, it starts releasing immature RBCs early into circulation, just to compensate for the low oxygen levels. All right. And I just added this for the sake of completion, but I don't think you need to know much about this. As we said, they have an average lifespan of 120 days. They lack a nucleus and organelles, and they rely on anaerobic respiration due to the lack of mitochondria. After 120 days, they are taken up by macrophages in the spleen and in the liver. And because they don't have a nucleus, if they are damaged, they cannot repair themselves and will die. Okay, so erythrocytes have a very distinct shape. They look like a ball with a dent in the middle. This is called a biconcave shape. And why is this important? Because this shape gives us a high surface area to volume ratio. Why do we need a high surface area to volume ratio? The red blood cell's main function is to transport oxygen, right? And to transport that oxygen very efficiently, we need to have a high gas exchange or a very efficient gas exchange. And this shape allows it to do this. Also, this shape gives it its deformability feature, which means red blood cells can squeeze in small vessels without getting damaged. And this deformability feature depends on two things, MCHC, which is the mean cell hemoglobin, and cytoplasmic viscosity. The cytoplasmic viscosity depends on MCHC. So if MCHC increases, cytoplasmic viscosity increases. But we need to keep this is why we need to keep the MCHC in very narrow ranges. Because if we do increase it and we increase the cytoplasmic viscosity, the red blood cell is going to lose its deformability feature, which it needs to carry out its function very efficiently. So we increase MCHC, we increase cytoplasmic viscosity, and the red blood cells become damaged as they squeeze through narrow capillaries or the splenic pores. And this is seen when red blood cells age. As we said, when RBCs only live for 120 days, so when they approach their 120 days, they lose their surface area to volume ratio. There's going to be less efficient gas exchange. MCHC is going to increase. Cytoplasmic viscosity is going to increase. And therefore, deformability is going to decrease. Lastly, they're going to get damaged by this because they can't squeeze through the splenic pores or the vessels. So they're taken up by splenic macrophages. And that is the first part of the lecture. Any questions? Are we all good? Okay. So now onto the RBC membrane. The RBC membrane has carbohydrates, proteins, and lipids, but there's a difference to this membrane compared to other membranes. It has equal parts of cholesterol and phospholipids. Why is this so important? Because as we mentioned, RBCs have a deformability feature, which means that they need to maintain this feature. How do they maintain it? By having equal amounts of cholesterol and phospholipids. If, let's say, I increase the cholesterol, I will increase the cell membrane strength, but I will also decrease its elasticity, which makes it lose its deformability feature. And this is linked to RBC abnormalities such as acanthocytes and target cells. The only reason I'm mentioning acanthocytes here is because the doctor did mention it in your slides, and the red blood cells will look like this. They'll look a bit spiky. So this is why it's important for the ratio of cholesterol and phospholipids to stay maintained. So RBCs don't change their shape and become deformed and can't carry out their function. For the phospholipids, they're unevenly distributed throughout the membrane. So you have two outside and two inside. And their names are very hard, I will admit. They have two on the outer membrane called phosphatidylcholine and sphingomyelin. And on the inner membrane, they have phosphatidylethanolamine and phosphatidylserine. The only one I think you guys need to know is phosphatidylserine. This is the only important one here, I'm pretty sure. Why? Because as we said, when RBCs age, they get taken up by the macrophages in the spleen. But how does this happen? This phospholipid flips to the other side of the membrane, and it leaves its receptor on the outside. So when the splenic macrophages come, they can bind to this membrane and get taken up. So the splenic macrophages takes up the RBC and takes it out of the circulation. This happens when it approaches the 120 days. And what induces this? Inflammation and this protein that your doctor mentioned called the C-reactive protein, which is produced during inflammation. RBCs also have these glycolipids on its outer membrane, and these are the ones that determine your blood type, the carbohydrate-based antigens. RBCs also have a protective coat on the outside called the glycocalyx. The glycocalyx is negatively charged. Why is this important? Because if they're negatively charged and there's an RBC next to an RBC, they're going to repel each other. This is important because if they attracted each other, okay, they would rub against each other and cause mechanical damage. This also applies for the endothelium. Because they're negatively charged, they don't get mechanical damage from the endothelium. It also protects them against microbial attack. And that's it for this part of the membrane. So moving on, in aging RBCs, this is a recap. Can anyone do this recap with me, please? Who wants to volunteer? That's okay, guys. I'll do it myself. Okay, so phospholipids, the phospholipid that we talked about, which is the phosphatidylserine, flips to the outer layer. And by doing this, it exposes its receptor. So when splenic macrophages come, they can bind to this receptor and take up this RBC out of the circulation. What could induce this? As we said, inflammation and the C-reactive protein. And they also have the glycocalyx, which is negatively charged and protects it from mechanical damage, and the glycolipids, which determines the blood type. Now, for the RBC membrane, there's this picture in your slides, which scares me a lot to this day. The first protein that's mentioned, there's four structural proteins that are very important in maintaining the integrity of the membrane. The first one is spectrin. Spectrin has an alpha chain and a beta chain. These chains are arranged anti-parallelly like this. Can you guys see me? And it forms a dimer. You also have this band 3 protein. It's an integral protein. It's integrated into the membrane, and it is an anion exchange pump. So it exchanged Cl for HCO3, which is bicarbonate. You also have this protein right here called ankyrin. It binds the spectrin spectrin, and it also binds band 3 protein. By binding to both of them, it attaches the spectrin to the membrane. And lastly, you have actin. Actin is bound to 4.1 protein and the spectrin tail, and it basically stabilizes it by doing this. It attaches all of it to the glycophorin C, which is an integral protein. So what do we need to know about this? The RBC membrane, as we said before, is very important because we need to maintain its deformability, and we need to make sure that it maintains its shape so it can carry out its function very efficiently. So we have the vertical interaction and the horizontal interaction. in the integrity of the membrane is maintained by the proteins band 3 and the other integral proteins. The horizontal interaction is maintained by the spectrin protein mostly and the other cytoskeletal proteins. And this is a very commonly asked topic on this topic, so make sure you know this well. Again, a recap of everything I said, so you guys can refer to this later. And as we said, the vertical integrity is maintained by the ankyrin and the transmembrane proteins, and horizontal is ensured by the spectrin and cytoskeletal proteins. Now, membrane defects. Sometimes the integrity isn't maintained. Why? Because you get defects in those proteins maintaining the vertical integrity or horizontal integrity. One example of this is hereditary spherocytosis, which is caused by the defect in the vertical integrity of the membrane. And it results in spherically shaped RBCs. This causes the RBC to lose its surface area to volume ratio, so it has a low surface area to volume ratio. And as they circulate through to the spleen and the rectal endothelial system, they face hemolysis and die. So these are the hereditary spherocytosis. You can see that they're very sphere shaped, and the doctor did mention this, and she said this is the only thing that you need to know. Other things were mentioned in your presentation, such as this table and this table. You do not need to know this. She mentioned that, and she said this was just there to show you that if you do change the ratio of the things in the membrane, this would result in this and changes in the shape of the RBC. And that's it for the second part of the lecture. Is everyone okay? Any questions? All good? What do you mean exactly by cytoplasmic viscosity? Okay, hold on, let me go back. Hold on. Where was cytoplasmic viscosity? I can't find it, but oh, I think it's here. No, all right. Cytoplasmic viscosity is the thickness of the cytoplasm. If I increase the MCHC, I will increase the viscosity of the cytoplasm. I'll increase its thickness. It will become more thick, which deforms the RBC. Is that okay? Okay. May you repeat what you need to know from the deficiency? The membrane defect, right? The hereditary spherocytosis. Okay. It's caused by the defect in the vertical integrity of the membrane. So when there's an issue in the vertical integrity of the membrane, the red blood cell loses its surface area to volume ratio and can't carry out its function very efficiently. And the doctor only mentioned hereditary spherocytosis, and she said you don't need to know the others. So she mentioned a lot of stuff, a lot of abnormalities, and a lot of pathological changes seen in the erythrocyte morphology. You don't need to know this. She just put it there as an example for you guys to understand that if you change this, this will change. No problem. Okay, moving on to erythrocyte metabolism, which is my favorite. I enjoy this one. But before we get into it, let's just talk about hemoglobin for a second. Hemoglobin is the molecule that carries the oxygen in the RBCs, right? It's made up of heme and globin. Heme contains an iron with a porphyrin ring. This is the iron, and this is the porphyrin ring. And globin is made up of 4 polypeptide chains, 2 alpha and 2 beta in adult hemoglobin, and they are made in the ribosomes of RBC precursors. RBC precursors because our RBCs don't have ribosomes, they don't have organelles. The heme is synthesized in the mitochondria, and they both meet each other in the cytoplasm to form hemoglobin. And these are the types of hemoglobin. I'm pretty sure you guys are familiar with this from the other lectures, but I'm adding it for the sake of completion. Now let's start the actual metabolic pathways. So we talked about how RBCs have a very distinct shape, and it has its deformability feature that allows it to squeeze through capillaries, right, without getting damaged. So we need certain chemicals to maintain its shape and its function so it could carry it out efficiently. What are these 3 metabolic pathways? We have the Embeden-Meyerhof pathway, the Lewberg-Rapport shunt, and the Hexose monophosphate pathway. Let's look at an overview of all the pathways together. This is the main one, which is the EMP pathway, or the anaerobic glycolysis. It's the first one that we talked about, the Embeden-Meyerhof pathway. and it's the main one that produces energy. And we also have the hexose monophosphate pathway and the Lou-Berg-Rappaport shunt. Okay, as you can see, all these pathways are integrated, and you'll see what I mean in just a second when we come back to this. The most important one here is the EMP, or the anaerobic glycolysis. Why? Because it produces the energy for the RBC. It takes in glucose by facilitated diffusion, and it converts it to lactate. By doing this, it generates 2 molecules of ATP. Why do we need these 2 molecules of ATP? Can anyone tell me why? Anyone, why do we need ATP? For energy. Yes, but why do we need that energy? Movement of RBC's metabolism. Okay. As we said, RBCs need to maintain their shape, right? For them to carry out their function. Or else they'll be like hereditary spherocytosis, the spherocytosis RBCs, and they won't be able to carry out their function efficiently. So we use this energy to maintain the cell volume, shape, flexibility, and the cation pumps. All right, you'll see what I mean by cation pumps towards the end. But are we good with this right now? Okay. It also generates NADH, but why do we need this NADH? When hemoglobin releases the oxygen molecule, the iron that we talked about that's present in the heme group changes from Fe2 plus to Fe3 plus. Why is this significant? Because the Fe2 plus is the hemoglobin form, and the Fe3 plus is the methemoglobin form. The methemoglobin form is non-active. It's not functional. It results in reduced oxygen transport capacity, which is really bad for us because we need this oxygen. We need RBCs to carry out their function. So what do? Remember the NADH that we just talked about? It's going to reduce methemoglobin to Fe2 plus. So it's going to give its hydrogen to Fe3 plus and reduce it to Fe2 plus. So it becomes the active hemoglobin and it can take up oxygen. This is catalyzed by the enzyme, which its name is very fitting for this process, methemoglobin reductase enzyme. This enzyme, from the name, catalyzes the reduction of methemoglobin. This enzyme is also called cytochrome C reductase. The doctor mentioned this, so again, I'm adding it for the sake of completion. So, can anyone tell me what NADH is used for? You guys need me to repeat anything because I know I'm a bit fast. Fe3 plus to Fe2 plus. Yes, I can repeat. Hold on, let me go back. Okay, so we said that the Embden-Meyerhof pathway produces energy. It produces 90 to 95% of the energy needed by RBCs. One glucose molecule enters the RBC by facilitated diffusion and is converted to lactate. By doing this, it generates 2 molecules of ATP. And we said we need this to maintain the RBC shape and volume so it can carry out its functions. And the NADH is needed to convert Fe3 plus to Fe2 plus. Why? Because Fe2 plus is converted to Fe3 plus when oxygen is released from the molecule. When it's released, it gets converted to this non-active form that we don't like because it has a reduced oxygen capacity. So we want to convert it back into hemoglobin. How do we do this? By the NADH that's produced from the Embden-Meyerhof pathway. It gives out its hydrogen and reduces Fe3 plus to Fe2 plus, making it active, and it can take up oxygen again. And it does this by the enzyme methemoglobin reductase, which is also called cytochrome C reductase. Are we good? Everyone good, guys? One last time, please. Okay. I can repeat, I don't mind. Okay, so remember the iron that's in the heme group, the iron that carries the oxygen, when it releases the oxygen to the tissues, it's carrying the oxygen, right? When it releases it to the tissues, it gets converted from Fe2 plus to Fe3 plus. Why is this very significant? Because when it's converted to Fe3 plus, it's converted from hemoglobin to methemoglobin. And methemoglobin has a reduced oxygen transport capacity. So it can't transport oxygen that well. And the main function of RBCs is to take up this oxygen and then give it to the tissues. So how is it supposed to carry out its function? we need help from NADH. NADH is going to convert this methemoglobin to hemoglobin by giving its hydrogen. This is catalyzed by who? Methemoglobin reductase enzyme. It's also called cytochrome C reductase. Are we good, guys? I'll repeat it as many times as possible. I don't mind. Okay, I think we're good. Okay, so as we just mentioned, yep, okay, no problem. Now onto the second pathway, the Lou-Berg-Rappaport shunt. What does this do? It generates 2,3-diphosphoglycerate. What is this and what does it do? This molecule reduces the affinity of hemoglobin to oxygen. Why would we want to do that? Because when hemoglobin is carrying the oxygen, we want this oxygen to be released readily to the tissues. So it's releasing it to the tissues that need it the most. In other words, it's helping the oxygen be released more readily because we're decreasing the hemoglobin's affinity to oxygen, so oxygen can be released into the tissues. And this is important in two cases that your doctor mentioned, which is hypoxia and acid-base imbalance. Hypoxia, as we mentioned before, is the insufficient oxygen supply to the tissues. In hypoxia, we need oxygen to be released more readily to the tissues. So 2,3-DPG or diphosphoglycerate accumulates and it lowers the affinity of hemoglobin to oxygen, so it can be released more readily. This is also seen in the acid-base imbalance because let's give an example of respiratory acidosis. I know, scary word. You'll take more of it later. But basically, in respiratory acidosis, you have accumulation of CO2. So your body needs more oxygen. So what happens? 2,3-DPG accumulates again, reducing hemoglobin's affinity to oxygen so that it can be readily released. All right? Any questions? I know the pathways are a bit much. Okay. Now, this is also very significant in the unloading and the loading of the oxygen. As each molecule of oxygen is loaded onto the hemoglobin molecule, it changes the shape of hemoglobin. How? The globin chains move on each other. So imagine these are the globin chains, okay? They move on each other like this, holding that oxygen, all right? It stabilizes the molecule so it can hold the oxygen. When it approaches the tissues that need it, the beta chains are pulled apart, 2,3-diphosphoglycerate enters, decreases hemoglobin's affinity for oxygen, and then oxygen is released more readily. All right. All right. And this was also mentioned in your slide, so that's the only reason I'm mentioning it. But high concentrations of H plus, yes, I can repeat, no problem. In the loading and the unloading of oxygen. So let's say I'm collecting this oxygen, all right? The chains move closer to each other like this. Can you guys see me? The chains are moving closer to each other like this, holding the oxygen, all right? When it approaches the tissues that need it, the beta chains move apart from each other, allowing 2,3-DPG to enter, decreasing hemoglobin's affinity for oxygen and releasing oxygen more readily. All right? All right. So high concentrations of H plus, carbon dioxide, and when the hemoglobin molecule is deformed, like seen in sickle cell anemia, the affinity of hemoglobin to oxygen is reduced. All right. Okay. Now onto the last pathway, my favorite pathway actually, the hexose monophosphate pathway. So this drawing right here shows you the normal pathway, the anaerobic glycolysis, the EMP, right? But in hexose monophosphate pathway, glucose 6-phosphate is converted to 6-phosphoglutamate. All right. When it's converted to this, it uses the enzyme G6PD, glucose 6-phosphate dehydrogenase. It needs the help of NADP plus, which takes up the hydrogen molecule and becomes NADPH. All right. This is very, very, very important because the hexose monophosphate pathway produces this NADPH. Why do we need this NADPH? NADPH is very important because it reduces oxidize glutathione to reduce glutathione. What's glutathione? Glutathione is a very, very, very powerful oxidizing agent that protects RBC from oxidative stress. That was a lot at once, so let's rephrase it again. Glucose 6-phosphate is converted to 6-phosphoglutamate through the action of which enzyme? G6PD, which converts NADP plus to NADPH, producing NADPH. This NADPH used from this conversion is used to convert oxidized glutathione to reduce glutathione. So basically, we're doing all of this, producing all of this NADPH, converting this to that to produce NADPH to convert the oxidized glutathione to reduce glutathione so that we can protect RBCs from oxidative stress. Oxidative stress from who? Reactive oxygen species. Reactive oxygen species are ions or molecules that injure RBCs, okay? And by using glutathione, we can neutralize its effect and protect our RBCs, all right? I'm going to repeat it again, and you guys are going to see it in text, but I just need to make sure that you guys got it here. Okay. So this is a very important enzyme. Can you please repeat a bit slower? Yes, I can. Okay, let's go back. So normally, glucose is converted to glucose 6-phosphate and then goes through anaerobic glycolysis to produce ATP, right? And the hexose monophosphate pathway, this glucose is converted to 6-phosphoglutamate through the action of the enzyme G6PD. This enzyme converts NADP plus, or with the help of the enzyme, we need NADP plus, which takes the hydrogen from glucose 6-phosphate and gives it to NADPH. We need this NADPH. Why do we need this NADPH? We need it to convert oxidized glutathione to reduced glutathione. All right? Do we have it here so far? Okay. So reduced glutathione is a very powerful oxidizing agent. What does that mean? It means that it neutralizes reactive oxygen species, which are species that are very harmful to our red blood cells. So we need to protect them from that using this reduced glutathione. Okay? Okay. Now, sometimes this very, very important enzyme, which is G6PD, is deficient sometimes. And this is very bad because if it's deficient, then we can't carry out this pathway and we can't produce NADPH, which converts this to this. Do I have to memorize the full name? The full name of, listen, you need to know G6PD, glucose 6-phosphate dehydrogenase, because it's going to be mentioned a lot in your other lectures. And NADPH is what's generated from this pathway. So yeah, we do need to know this, unfortunately. Okay, now, when this enzyme is deficient, that means this pathway is deficient. And we can't produce NADPH, and it can't reduce oxidized glutathione to reduce glutathione. So we're going to have an increase in, we can't neutralize the intracellular oxidants, which are the reactive oxygen species. Therefore, the hemoglobin that's in RBCs denatures and precipitates and forms something called Heinz bodies. These are Heinz bodies. Let me show you. These are the Heinz bodies, as you can see. The hemoglobin denatured and precipitated because we couldn't produce the oxidizing agent glutathione. And because we couldn't produce it, we couldn't protect our RBCs from the oxidative stress. What does phosphoglutamate do? Is it important for us? Phosphoglutamate, this one you're talking about, right? It's not important. You don't need to know anything about it, but the reason it's converted from this to this is to produce NADPH. That's what we need from the hexose monophosphate pathway. We need NADPH. The doctor mentioned it in your slides a bit much, so that's the only reason I added it, and also to show you that's how NADPH is produced. Are we good so far? Okay. This is everything I said put into words again for your own reference. I mentioned how everything's produced, and you could also refer to drawings later on. Okay, Now, recap of the 3 metabolic pathways. Who wants to do this with me? I know it's been a lot, but we're almost done here. Any volunteers? Okay. Can you open your mic, please? Okay, so the first pathway that we had was the Emden-Meyerhof pathway, right? Okay.
Okay, sorry, my mic was off.
Okay.
I'll try.
So the first pathway we have is the Emden-Meyerhof pathway, right?
Yeah.
Okay, it produces, oops, sorry guys. It produces 2 things. What's one thing that it produces?
2 ATP
Correct. Why do we need those 2 ATPs?
to maintain the deformability and flexibility and cation pumps.
Correct. And we also generate something else from the Emden-Meyerhof pathway, which is?
Not sure, but is it NADP?
NADH. It's
NADH.
important to
Okay.
Yep.
All right.
And what does NADH do?
It reduces Fe3 to Fe2.
Correct. So it reduces methemoglobin to hemoglobin. Our second pathway is the Luberg-Rappaport shunt. What does this shunt do?
It generates the 2,3-DPG, which reduces the hemoglobin affinity to oxygen.
Correct. And our last pathway is the hexose monophosphate pathway. Which one? What does this one do?
This generates NADPH through the G6DP, and NADPH reduces GSSG to GSH, and GSH is needed to neutralize the oxygen species.
Yes, the reactive oxygen species. Correct. Thank you. Thank you, Lean.
You're welcome.
Okay, now on to the last part of the lecture, the metabolic defects. So, enzyme defects in glycolysis would result in decreased oxygen production, right? And this would be very bad for the RBCs because now they can't maintain their shape or they can't carry out their function efficiently. So this would result in hemolysis because they can't maintain their shape, they can't maintain their deformability feature. And so when they squeeze through capillaries or through the sphenic macrophages, they will become damaged and it will result in hemolytic anemia. The doctor mentioned 2 very important enzymes that are in glycolysis, but she didn't mention glycolysis anywhere else. So I'm only adding this for the sake of completion. But I don't think you're going to be tested on this because pyruvate kinase deficiency and phosphoglucose isomerase deficiency is going to be taken later on in mole. But that's the only reason I mentioned it here. All right. And she also mentioned this very, very, very scary picture in your slides. You don't need to know this. She mentioned in the recording that this was just there to give you an overview of everything. It's very scary, has a lot of enzymes, a lot of pathways, but you don't need to know much from this. Now, remember how we talked about Fe3 plus being converted to Fe2 plus? Sometimes this very lovely enzyme isn't there. And when it's not there, we get this, when we get this disease or this thing known as methemoglobinemia, which is too much methemoglobin in the blood. And this can be caused by the reactive oxygen species that we just talked about, that we're trying to protect from RBCs. They can oxidize Fe2 plus to Fe3 plus. This can also be caused by the deficiency of the enzyme that we were just talking about, the methemoglobin reductase enzyme or the cytochrome C reductase. Or your hemoglobin could just be structurally abnormal, and so it causes this. You can also get it through drugs or toxic substances that oxidize hemoglobin. And if this occurs, the patient will appear blue. They'll have cyanosis because hemoglobin can't transport oxygen anymore. It doesn't have a normal the transport capacity. It has a reduced oxygen capacity. So the patient will show cyanosis. Now, onto the last part, osmotic balance. RBCs maintain their osmotic balance. How do they do this? Why do they need to maintain their osmotic balance? Because they carry hemoglobin, this causes an osmotic pressure. So that means water could move in, but we need to stop water from moving in so they don't burst. So how do we do this? Through the membrane selectivity. So it chooses what it wants to bring in. It chooses if it wants to bring in sodium or potassium or chloride, or through the aquaporin channels and the sodium potassium pump. What does the sodium potassium pump do? It takes out 3 NAD and brings in 2 potassium, and that maintains the normal equilibrium of the RBC, all right? And it also has the calcium ATPase, which maintains low intracellular levels of calcium. These two, the sodium potassium pump and the calcium ATPase, need ATP. The aquaporin does not. It's just a channel that allows water to come into the cell. But because these are maintained and they maintain this concentration, this osmotic equilibrium, water does not come in. And we also have ion channels that we talked about and transporters. The transporters, such as the one that we just talked about before, the band 3, which is the anion exchanger that exchanges Cl for carbonate. And this maintains the pH in the RBC, all right? But let's say I didn't have ATP. What would occur? The sodium potassium ATPase wouldn't work. The calcium ATPase wouldn't work either. So calcium would come in, sodium would come in, and following this would be water. And it will all rush into the RBC. This would result in water accumulating in the RBC, and the RBC will swell up and eventually burst. This is known as osmotic hemolysis. And in sickle cell disease, when the hemoglobin has a what is it called? When you exchange valine, when you exchange it, it causes the hemoglobin to change its shape, right? And therefore it changes the RBC shape, right? Does the aquaporin also allow efflux of H2O from the RBC? No. It allows the entry of water. But because we're maintaining the normal osmotic equilibrium using these two pumps, it doesn't come in. But if we lose ATP and these pumps don't work, sodium comes in, calcium comes in, and because of this, water follows. All right? All right. And in sickle cell disease, when there's a mutation that occurs and then the hemoglobin polymerizes, RBC changes into a sickle shape, and then the membrane becomes more permeable to calcium. So again, calcium comes in, water comes in, and it results in hemolysis. Can you repeat the point about band 3? Sure. This one right here. Band 3 is the protein that we mentioned in the beginning, and the ones that maintain the membrane integrity. Okay. Band 3 is an anion exchange pump. It exchanges Cl for bicarbonate, which maintains the neutrality of the RBC and the osmotic balance. All right. Okay. So now on to questions. Who's gonna go first? Let me choose someone before I open the questions. Who wants to go? Okay, I'll open up the question and then see who wants to go. In which of the following is the extravascular hemolysis of RBC most likely to occur? E, correct. Correct. Anyone else that doesn't say E? Okay. Recognition of damaged RBCs by splenic macrophages happens as a result of which of the following? Okay, can you explain why? Okay. So remember how we said in the beginning, the RBCs only live for 120 days, and by the end of the 120 days, they get taken up by splenic macrophages. Why do they get taken up by splenic macrophages? They get taken up by splenic macrophages because they lose their deformability feature when they approach 120 days. The MCHC increases, the cytoplasmic viscosity increases, and so the RBCs don't have that deformability feature anymore. And when they come to the spleen, they can't go through the splenic pores, and they get taken up by the macrophages of the spleen. And this is the most likely thing to occur because all the RBCs after 120 days go through this. Okay. Now, recognition of damaged RBCs by splenic macrophages happens as a result of which of the following? B, 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 B. Yeah, it is B. Good job. Okay. NADH is needed for protection from oxidative stress, conversion of methemoglobin to hemoglobin, conversion of GSSG to GSH, or production of 2,3-diphosphoglycerate. Okay, I see B, I see C. Does anyone want to open their mic for this and tell us the reasoning behind their answer? Anyone? I see B and C. Okay, Dean, you can go.
Oh, okay. So NADH converts methemoglobin to hemoglobin, which is Fe3 to Fe2, and NADPH converts GSSG to GSH.
Correct. Very well done. And that's the end of the PAL session. I know, is it C? No, it's not C because that's NADPH. All right. That's the end of the PAL session, guys. I know I'm a bit fast, but bear with me. Could you please repeat why it's B? Sure. So, as we said, NADH is needed for the conversion of methemoglobin to hemoglobin. NADPH is needed for the conversion of GSSG to GSH. So make sure you guys know the difference between them because they're very confusing. All right. There's another question. Hold on. In the first question, what if the question said intravascular? What would be the answer? So intravascular hemolysis of RBCs. So RBCs dying inside the blood vessel. That would be RBCs entrapped in the blood vessels. I think. I'm not sure. Any other questions? No problem. Thank you, guys.